Welcome to our final segment where we will focus on future directions for heavily pretreated patients. We see our currently available bispecifics and CAR-Ts being evaluated early in the treatment course and in novel combinations. However, we will focus on where the field is moving and some of the new agents. So before we talk about new agents that we may not have talked about, one of the things that is starting to emerge is combination therapy with bispecifics. So he says, what's your take on the data so far on combinations of bispecifics, two bispecifics, bispecifics plus CELMoD, bispecific plus CD38 antibody? What's your take?
Yeah, no, I think the data looks quite fantastic, actually, especially with antibody combinations and two bispecifics for sure. But I think my hope is that what that data is going to show us is that by combining, we can get even quicker responses that hopefully will allow us to do limited therapy of the bispecific. And I hope that that's where we're heading. But I think the data that's really wowed me the most is the redirect data with the combination of talquetamab and teclistamab, showing these impressive results, especially in the extramedullary disease patient.
So what has stood out for you, Gurbaksh?
I'm very excited for the combination therapies. I'm particularly interested in where there's a combination of an IMiD in addition to a bispecific, because one thing we're seeing in myeloma is that we think, well, we can give all the immune therapy that we want, but what's happening to the immune system? Is the immune system fit enough to actually tolerate, to keep on going, right? Are the T cells exhausted? And I think we know some actions that the mechanism of action of how the IMiDs work or the new CELMoDs work. I'm sort of, I think mechanistically, I'm very interested by that. Even the TRIM2 data, where you have a combination of, I believe it was DARA plus TALC plus POM. That's impressive data. I think a median time to first response in that trial was 1 month, and the median PFS was 15.5 months, and the responses were durable. And this was a heavily pretreated patient population, actually. The median prior lines of therapy was 6. While redirect is nice because you can have two bispecifics, I think the IMiD and the bispecific combinations, I like that quite a bit.
So Caitlin, we've heard about two bispecifics. We heard about IMiDs with bispecific. There's some data on CD38s with bispecifics, and the rationale there being not only that it is going to be hitting the myeloma cell in two different ways, but perhaps modulating the environment. So what's your take on that combo?
I mean, we as myeloma docs are no strangers to combinations, right? I mean, we got so used to drug approvals being with 30% overall response rates. And now the bar has been thrown down where we have these high, response rates of 60, 70% with our bispecific. So we hope that any combination leads to improved or overlapping or sequential successes, as long as we do not have overlapping toxicity. So we are really excited to see data like what you're talking about, combining the bispecifics with DARA, for example, or with IMiDs, whatever that may be, and really seeing very high overall response rates, but also starting to see that we're trying to kind of remodel the immune system to enhance perhaps the drug responses, as long as we are not putting these infectious risks that much higher. There were deaths seen with these patients getting these combination therapies with DARA, for example, and we have to be very careful to make sure that we are protecting these patients while offering them kind of the next novel combinations.
So Christina, what's your take on the pros and cons of the combinations?
Well, it makes sense, right? Because like Kathleen said, the responses are so high, almost unbelievable in this population of patients. We went from, selinexor giving us less than 30% response in the pentarefractory to these 70% responses. Some of the combination make absolutely sense. Like my colleagues mentioned, the mechanism of action of the IMiDs. Can we improve the T cell fitness, right? Can we use them sequentially in combination? A lot of clinical trial of using maintenance post-CAR T cell with the same concept of improving the T cell fitness. So it makes absolutely sense to me. And my concern, like everybody else, we don't want to, unfortunately lose in toxicity, because we know that we have some patient experiencing myelosuppression. Now we are adding an IMiD, for instance, the berdomide, Medzi, can we cause more myelosuppression, more risk of infections? So we have to be very cautious. But, and myeloma has evolved now for the last 2 decades in that direction. We started with a couple, 3, 4, and so it makes sense. And these drugs are going a little bit everywhere, including in the maintenance post-transplant. And I have a lot of questions about that. What we're going to do next at time of relapse when they're being on this bispecific for 1-2 years. We were talking about sequencing these drugs. So we're opening a can of worms. So we're going to have maybe, some innovative combinations, but we have to be cautious about, the sequence, increasing the toxicity. So it's a lot to say.
Abdullah, what's your take on combo therapy?
Yeah, I have to echo all my colleagues. We definitely know how effective the bispecific as a monotherapy. We know the effectiveness is like in a complete response over 40%, and how much the median duration of response for these patients. It makes a lot of sense, as Dr. Caitlin say, like we're gonna do a combination. We know that. Which combination is better? Is it with 2 bispecific BCMA and GPRC5D, which I think we will see that, like the redirect therapy trial, or a bispecific BCMA with daratumumab CD38, or with an IMiD or a CILMOD. I think these combination therapy are fair enough to actually see it in using. We will see the efficacy. There's no doubt about it. I think we'll see better results, deeper response. But I think the biggest fear that all the colleagues agree with is the toxicity. So can we prevent the toxicity by achieving a great remission and have a fixed duration of treatment? I think that's going to be the biggest question. And we're hoping that these trials are going to be designed to, hey, if I got the patient in MRD negative, which I think it's not going to be very difficult, due to the fact that these initial, preliminary results are showing really impressive efficacy, can we stop the drug or decrease the frequency immediately and decrease the side effects so that the side effects are manageable and we can apply these treatments to our myeloma patients?
Great. So moving on to another topic. So Christina, we have CAR-Ts in development to other antigens.
Yeah.
You excited about anything you've seen so far?
Yes, I am about, of course, the GPRC5D CAR T-cell for sure, particularly because, looking at the data, the toxicity that you see with talquetamab are very limited. They're not protracted. And so I love that, the fact that it's a very effective CAR T-cell, a different target, and the toxicity surprisingly is limited to the earlier part, but it's not like a lingering toxicity that we see with the talquetamab continuation. So I'm very excited about that. And of course, the dual CAR T-cell targeting 19 and BCMA that we're all talking about, very excited about preliminary data, and also the allogeneic CAR T-cell. I'm part of one of the clinical trial, and I was able to review some of the data. I like it, it's effective, it's not as like, the anticipation, and the fact that, we have the availability, the universal donor. So some of these patients, they can wait for the manufacturer, for the autologous CAR T-cell. We can definitely overcome that obstacle. So very intrigued since the very beginning from the allo CAR T-cell, and I was excited to see that the preliminary data are actually quite good, quite outstanding.
So Abdullah, other targets, dual targeting, allo CAR Ts, perhaps, fast CAR Ts one day. Where are you excited?
So I think I agree. I think the good thing about the allo CAR T, I think the concept, it makes a lot of sense, using other people's CAR T in order to have a better efficacy and less side effects, but, are we talking about the efficacy as equal as what we have currently in the CAR T? I think it's going to be very difficult for us to kind of manage it. Now there's a lot of companies, and I'm sure, like some of my colleagues are aware about that. There's companies that are trying to get the CAR T manufactured within a week, and some of them within a days. So I think that the days of waiting for the manufacturer is about to go away, and that we will have, hopefully, I don't think we will need to talk about bridging therapy for these patients. So you get their CAR T apheresis and then you'll get it back. So I think these type of CAR Ts are still, with that efficacy and having the manufacturer will be available. The question here is that what will be the allo CAR T? Where do we put the allo CAR T in this situation? I think that allo CAR T maybe have to be studied more post CAR T and BCMA. See how much is it effective, how much it can actually provide efficacy and duration of response. Because I think that's one of the trials that actually was open in that case. So other targeted therapy, the GPRC5D CAR T. This is really the phase 1 study showed really impressive results. And I think it makes a lot of sense to have this type of CAR T to focus on that. I'm hoping that we will not have that side effects that we're seeing with talquetamab, or if we have it, it's minimum side effects, because I think that will also play a big role to actually choose this type of CAR T in our myeloma patients.
So Gurbaksh, what's your take on all the new data coming down? Some new BCMA CAR Ts, some by-targeting CAR Ts, some allo CAR Ts. So
I think the data is all quite exciting, because I feel like, the CAR T journey in myeloma, this is just the beginning. One of the things that, we have to achieve is that, we want to have the deep and durable responses that we're seeing with some of the CAR Ts. But we also want to limit some of the other toxicities that we see, such as the neurotoxic effects, right? CAR Ts. And I think especially like ddBCMA, I'm quite excited about that, especially knowing its efficacy, and then not seeing as much of the MNTs or the Parkinsonian type of features that we're seeing with cilta-cel. So that's exciting. I'm also in agreement with Christina that the GPRC5D as a CAR T drug target, I think once it gets approved and makes it to the myeloma market, I think there will be a much more acceptance of it within the myeloma community, because you are not seeing the same oral side effects. And so all of this is exciting. I think we're moving in the right direction. It's just to see how things will pan out.
Jesus, you got a favorite?
So I think this whole time, right? So CAR Ts, the one and done concept, has always been intriguing to all of us. And then you have the bispecifics that are available right away, but you have to give them continuously, at least for now. So the allo CAR T has always been the holy grail, right? Where you would have that off-the-shelf, one and done kind of therapy. But we'll have to wait and see if they're going to be as efficacious as the autologous CAR Ts. I like the dual targeted CAR Ts, or potentially even triple targeted CAR Ts, because for the first time we can combine CAR Ts, right? We talk about combination with bispecifics. CAR Ts are hard to combine with, but I think that's the way to try and kind of do a combination therapy within a CAR T.
Caitlin, you get the last word on this?
Last up, I mean, it's hard. Everyone has said such wonderful things. So just to, not to echo everything they've said, I agree, but just to think outside the box a little bit and take it a little different direction. There are wonderful novel things coming, but we also have some good old faithfuls we can go to here with our cilta-cel or ide-cel, and looking at using them in different aspects of our disease care, right? So lots of studies looking at how we can use CAR T earlier on in the process, whether that's newly diagnosed, randomized fashion, comparing it to auto transplant for consolidation, looking at using CAR T as consolidation, again, perhaps for those patients who have functional high risk, relapsing soon after auto transplant. So I'm really excited to see where we can kind of throw all our CAR Ts against the wall and figure out where it sticks best.
So another group of drugs that has our attention is the CELMoDs, the cereblon modulating drugs, and there's some other even protein degraders coming down the pike that are targeting the cereblon molecule and the downstream effects. So Jesus, what's your take on these new IMiDs?
Yeah, no, I think those are great. I mean, I think, way back when thalidomide, lenalidomide were first approved, we didn't really know how they worked. And then eventually it turned out that they work by inhibiting cereblon, right? So now that they can directly do so, I think you're getting so much less scatters. So the toxicity profile is so much better with this new CELMoDs than the old IMiDs. And I think that's important, right? We all have patients who are lenalidomide maintenance that are having diarrhea and fatigue and all these things that usually leads to them discontinuing. So my hope is that I think this new CELMoDs will be much more amenable to something like a maintenance or the combinations with less toxicity. I think it's unfortunate they're coming in the heels of all these incredible therapies we're talking about with response rates of 60 to 90%. The approvals are getting caught in that because obviously the single agents are not going to be that powerful, but I think they have a lot of potential in combination.
So Gurbaksh, where do you see CELMoDs fitting into the equation?
I think it would be naive to say if we know the place, to be honest, because the landscape changes. 3 months ago, if you had told me I'm going to say it's going to be, I probably think it's going to be post-CAR T relapse. That's where I see it fitting, just the way historically it's been studied, right? But it's the treatment landscape changes so often. I would like to sort of my personal preference would be like if we do one immune therapy, such as the CAR T, and then there's a relapse. I would like a change of strategy and possibly a CELMoD in combination with something else used in that space, and then another immune therapy, just so the immune system kind of gets reinvigorated. I think that is ideally a place for me. Now, what happens in real world, time will tell.
So, Caitlin, do you have a crystal ball? Where do you see CELMoDs fitting in?
I think the thing I struggle with the most is this, hope for this treatment-free interval. And as much as I understand the interest of putting these CELMoDs in post-CAR T to kind of improve T cell persistence, and for that matter, I suppose you could say the same for bispecifics. There's lots of interest in combining it there as well. I hope the juice is worth the squeeze in that regard. I really hope that it'll be worthwhile, because otherwise we're taking away this kind of carrot that we're dangling in front of people that says you don't need any treatment. So I'll see. I think its intent post-transplant makes great sense to me. I hope that it is worth it if we do it with the T cell redirectors.
Christina, so have you had the opportunity to use CELMoDs in trials? Have you followed the data?
Yeah. I have a couple of interesting trials, actually, combination with selinexor, with the mezigdomide, and also post-transplant, the iberdomide compared to the lenalidomide. While I agree, the treatment-free for our patient is going to become very important, and we are asking lots this question. A lot of the clinical trials are designed to determine can a certain point stop therapy, but it's mainly for the newly diagnosed. We know very well that when in the relapse setting is a different story. And so, for me, the maintenance post-CAR T cell or bispecific in the heavily pretreated patients makes more sense, because unfortunately, I have to say, we've done a lot of CAR T cell, commercial CAR T cell treatment at Duke, but patients heavily pretreated, they relapse within a year, a couple of years max for some patients. So then we have to start over. So if we found a way to prolong the duration of these responses for the heavily pretreated, why not? And I always love the IMiDs because they're by mouth, and it's true, we have cumulative toxicity, we have the risk of secondary malignancies, but at least in terms of quality of life, taking something by mouth with the possibility of remaining in remission longer. So I like the idea. I like that niche for some of these stronger CELMoD, and particularly in the heavily pretreated patients.
So Abdullah, we've certainly used the IMiDs a lot. Do you think CELMoDs are going to be there in the future in the same kind of numbers?
Well, I mean, I think, I have to echo all my colleagues and add a couple of things. First of all, I think we will not have the same problem to introduce the CELMoDs to our community oncologists, because they use a lot of Revlimid and pomalidomide. So that's going to be a positivity. However, all the initial studies, kind of like, although it's still earlier to kind of say that the iberdomide and mezigdomide was used in combination with Dix, I really think that we are hoping to see more of the studies. I know there's the Successor 1 and 2 that is combining the mezigdomide with a proteasome inhibitor, and there's other studies that combining iberdomide with daratumumab, and other studies to combine CELMoDs with bispecific. So I think that's where we're going to use this in relapsed myeloma, but where exactly? I think that is going to be a challenging question. Most likely, I think most of our patient population are going to receive it after they fail all the treatments. I think mostly after the BCMA GPRC5D. I will highly recommend, if I'm going to use it, it's going to be in a combination therapy with other drugs if that got approved.
Abdullah, let's start with you. What is your message to the community for the treatment of highly relapsed refractory myeloma? What the future holds for this patient? What is it that you're excited about? Anything we've not talked about?
Yeah, so I mean, first of all, as all my colleagues have say, the field of treatment of multiple myeloma changes a lot. Any treatment that get introduced and FDA approved, things change in terms of how we treat the algorithm. Any treatment that moved up front, everything has changed. So always my advice to my colleagues, my community oncologists, is remember to always keep in contact with our academic centers. We treat a lot of myeloma and keep up to date in terms of the newer treatment options for the myeloma patients. Always remember clinical trials is the reason why we're here right now. We are talking about survival benefit that improved years ago, prior to the thalidomide before 1999. We have all chemotherapies. Now we're talking about immunotherapy treatment, bispecific and CAR T cell, and it didn't happen for no reason. It happened because we had clinical trials. We always recommend, and advise our colleagues is always make sure that they are aware about all the clinical trials that are active in very near sites, and to encourage patients to kind of like, have access to that. My biggest thing I'm hoping in the future, two things. I mean, I know maybe we didn't mention that, but the cifostamab that targets FcRH5, I'm hoping that we're going to see this drug, because now we actually have the talquetamab and hopefully CAR T GPRC5D. We have to be a little bit, realistic. We're not curing myeloma, unfortunately. We need some other targeted therapy. We need more options, and I think maybe the CELMoDs will happen here, but the target of FcRH5 have really impressive results. And if this drug get approved, I mean, I think we will have a good option in the back of our pockets.
Well, that is very true. Glad you brought that up, because yes, we've seen some very convincing data even in BCMA failures with cifostamab. Thank you. So Christina, your parting words of wisdom for the future of relapsed refractory myeloma therapy for the community?
The reason why some of these drugs are available for our patients because went through clinical trials, and so always the possibility of participating to a clinical trial, very important. I also believe that in addition to risk-adapted therapy, response-adapted therapy, I also believe on personalized medicine, and I love also the evolution, development of some of these targeted therapy for a subpopulation of patients. We know the story with the venetoclax and with the more potent BCL2 inhibitor, the MMSET and clinical trial for the 414 translocation. I love that. Is another oral agent, but some of these patients and the RAS mutation, and there are so many under development. So personalized medicine, also understanding, the importance of risk stratify this patient, how we're going to treat the standard risk is different from the high risk disease. And we do have clinical trial from the very beginning. They're designed targeting this population of patients. Finally, I've been thinking about that since I started myeloma. Myeloma is not one disease, it's many diseases. So, again, I think is while the community physicians are doing a phenomenal job on treating these patients, is a collaboration. I say it again, I repeat myself, because participation in a clinical trial, how we can help patients to receive these drugs in the community, the best sequence, etc. The future of myeloma is still quite bright. I mean, so we do have the new drugs, the trispecific and the targeted therapy, the new CAR T cells. So we have a lot of new drugs under development. I'm very excited.
Well, thank you for bringing up all that. Yes, the BCL2 inhibitor venetoclax may have not made it in trials, but we still use it for our BCL2 positive 1114 translocation partners off label, and new BCL2 inhibitors and rotoclax and others are in development. And yes, targeted treatment and developing, algorithms for subsets of myeloma is certainly the way we have to, go. So, Caitlin, what is your, future for myeloma relapse refractory looking like?
Sure. I think broadly what I would say is that the kind of era of novel agents is kind of changing. As we are putting more and more of our drugs in earlier lines of therapy, triple classes are going to become more and more present earlier on in their disease course, where immunotherapy is kind of here to stay. My main goal for the future is response-adapted protocols, ways that we can meet our unmet need of myeloma is stopping therapy for these patients.
And Gurbaksh?
I think I would echo what Caitlin said. She always makes wonderful points. The future is bright. We do have lots of newer options on the horizon and different strategies, especially when there's, dual targeted CAR Ts, so we can limit the toxicity of these agents as well. But at the same time, I think that the unmet need in myelomas, there's a population of patients we may undertreat, and there's a population of patients in myeloma we overtreat. And I think it's about time we learn biologically how these patient profiles are different and how to tailor therapy for them. So I think in the future, there is going to be more emphasis on limited duration therapy or risk adapted therapy based on the depth of your response. And I'm actually most excited about that because quality of life, financial toxicity, all of those factors play a role. And we need to sort of meet that need for our patients.
So Jesus, as we sign off, what is your words of wisdom for your colleagues in the community?
So I would echo everything. I think the future is very bright for myeloma, and that means use your best available therapy always. Do not hold back or hold something new will be coming. So always use what's best at the time without worrying about what's next.
Well, thank you all for these very important discussions and considerations. It is really important for our patients and valuable for us to have these conversations. We want to thank again Sanofi and Janssen for their generous grant support and the Cancer Communicator for the program management and development of these activities. I hope you all enjoy the rest of your day. Thank you very much.